Whenever we are working onto the pharmaceutical product, we require the starting point for the dissolution method development. We can do the literature search and find out the dissolution method used for the particular product. We can go through the monographs in the pharmacopoeias. We can search the literature. We can check the journals. Also, we can check the methods generally adopted for the dissolution testing. And FDA Dissolution Method Database, it is also called as OGD database, provides the methods used for the dissolution testing. These methods are adopted by the innovator formulations, or you can say the novel formulation manufacturers or the NDA applicants, and from that the dissolution methods are taken into the database. This database is provided by the FDA. It is called as, or it is also known as OGD database, and you can get the dissolution method from this database. I welcome all the viewers to this very informative topic onto the OGD dissolution methods database. OGD is a tool for developing and evaluating the generic formulations or generic drug products. Also, you can refer this database for making the novel formulations, or you can use it for making the hybrid type of formulations. It can guide you to start the method development. Now see what is the OGD dissolution database. So it is developed by the Office of Generic Drugs by the US FDA. It is publicly available database, and it provides the FDA recommended dissolution methods for the approved drug products. So whatever the products are approved in the US market, the dissolution methods adopted by those formulations and the ND applicants are enlisted here. OGD database is important and essential for the ANDA submissions, generic formulation development, quality control and regulatory compliance. So always you can visit this database and get the information for the dissolution. Purpose of the OGD database: to standardize dissolution testing methods, to ensure bioequivalence and product performance, to help the developers to avoid in vivo bioequivalence studies when possible, and to support the biopharmaceutics classification system based bio waivers. This is the wide scope and purpose of this database. So whenever you are developing a formulation or developing a dissolution testing method, you should visit this OGD database and check the method available or the dissolution conditions available. So if you want to check, you should have the drug name, dosage form information. Then you can get the information onto the dosage forms like tablets, capsules, dissolution apparatus, rotation speed or flow rate, dissolution medium, media volume, sampling time points, and if USP monograph reference available. If you visit this database and if you see that the USP monograph reference is given, that means the method is already available or the parameters are already available into the USP. So directly you can go to the USP and check in the monograph. Various type of dosage forms are covered by the OGD Disso media or dissolution database. Tablets, various types of tablets are there: IR, ER, delayed release, enteric coated, chewable and ODT. Soft gels are there, capsules are there, suspension and injectables are there. Various different or differentiated formulations, novel formulations like transdermals, implants, vaginal rings, topical, rectal and ophthalmic products are given into the database. Apparatus, different type of apparatus are there. Apparatus 2 is the most common type of apparatus. Apparatus 1 and 2 are given. Other apparatus are used for the formulations like ER, transdermal and topical formulations. Non-compendial apparatus like shaker, sieving machines. Dissolution media. So this information is available onto the OGD database. Dissolution media pH range around 1.2 to 12. So includes both the physiological and non-physiological medias. Media types like deaerated water, buffer solutions, surfactants and their levels are given there like SLS 280. Enzymatic medias are there. Simulated GI and intestinal fluids information is available. Use of organic solvents is generally restricted, but some of the formulations are there in which organic solvents are used. So use of surfactants is given. Improved solubility of the BCS class 2 and 4 drugs. For this, the surfactants like sodium lauryl sulfate, UNAT, LDAO, CTAB, Triton X100. So all the surfactants you can find in the database. These are very product specific. Then you can also find the sampling time points for IR formulations. Typically 3 to 5 time points are there like 5, 10, 15 and 30 minutes. And early sampling might be there for 1 to 3 minutes for ODT type of formulations or tablets and the sublingual tablets. For ER formulations, the time points may range from zero point 0.5 to 24 hours, and in some cases, it may be for 96 hours to 40 hours, like implant formulations. Test conditions, media volumes are given, mostly 500, 750 ml, 900 ml, 1000 ml are given, but you can find in the OGD that 900 ml media volume is the most common media volume. And temperature ranges are given, or temperature is suggested for the dissolution. Mostly 37 degrees Celsius is suggested. For topicals it is 32 degree and others may be 25 to 45 degrees Celsius depending upon the formulation. Applications like formulation development, quality controls, specifications, regulatory submissions which may be ANDA or supplements, and supporting the IVIVC and BCS based bioweapon. So OGD has a wide application starting from early development till the IVIVC and biowaiver applications. Also regulatory submission, you can take the help of OGD. Now how to access this database? So you can type in the Google OGD database and directly you can be taken to the website access data fda.gov. So in the upcoming slides in this video I have shared the screenshots for your easy understanding. The database is searchable by drug name, dosage form, apparatus and the media types. The OGD database is a very comprehensive and important tool for method selection, regulatory alignment, ensuring the product performance, and that's why this database is highly recommended for the generic drug developers. Now see some of the important points for the FDA dissolution method database. The purpose of this database is to provide FDA recommended dissolution methods. So directly you can use these methods, but always know that you need to prove the discriminatory power of the dissolution method. Then it provides the dissolution method for the drug products which are not listed in the USP. So if you cannot find the monograph or the product is new and the monograph is yet to come, then directly you can visit this database and collect the information as required. It is especially for use in the generic product development. It is maintained by the Division of Biopharmaceutics under the Office of Pharmaceutical Quality of the US FDA. You can search the database using the generic name of the API or formulation. Like you can search by metformin and you will get the list of metformin products. Out of that, you can select the combination of metformin, or you can check the extended release type of tablet, type of formulation. So you can check your uh, formulation and then select the procedure given or the dissolution parameters given in the database. For combination products also, you can check by the alphabetical order, and you should not use any slash or spaces. So once you use this database, you can easily understand how to search. You can search by using the name of ingredient. You should avoid the spaces around the slash and reversing order, omitting the slash. Drug names listing in the search results. All drugs are listed alphabetically by the generic name. Combination products are sorted by the alphabetical order of the active ingredients. So in the dosage form section, you can get the information about the dosage form like tablet, capsule, suspension. Description of each USP apparatus is there, and the type of USP or type you can get from the USP. Similarly, type of medium is given there, and the preparation method you can take it from the USP. The testing is generally performed at 37 degrees Celsius. Now, numbers in the recommended sampling times. What is the meaning of this? These sampling times represent the sampling intervals in minutes, and otherwise if it is given in the hours for generating the dissolution profile. By the final time point, 70 to 85 Q, that is percent drug release of the formulation should take place, and the final time point is like that the formulation releases the 70 to 85% of the drug. And generally the date updated section is there, which provides you the date on which it is last revived, received, or revised. So then frequency of the updation is it is updated quarterly. It is updated in April, July, October, or January, for example, and by the 5th working day of each quarter. Here I have shown the image where you can check the dissolution method database. Here I am showing. Now you should go to the search, dissolution method database search. If you click here, you will be taken to the next page. This page will open. Here in a search for dissolution method, you can put the name of API and you can submit it. For example, I put here sitagliptin and I submit it. Then I will get this information. So in this drug name is there, dosage form is there, USP apparatus type is there, speed and RPM, medium, media volume or volume in ml, recommended time points, and the date updated. Then also one thing here we should know that if in the medium it is written that refer FDA's dissolution guidance 2018, that means you should refer this US FDA guidance for the dissolution method development. And generally I found this type of instruction that refer to FDA's dissolution guidance 2018 means the drug is highly soluble. It may be belonging to BCS class 1 or 3, and you need to refer the FDA's guideline uh, published in 2018, and from there you should take the uh, information like media, USP apparatus selection, speed, everything. So other type of information you can find here like dosage form. So extended release tablet or tablet it is written. If only tablet is written, that means it is a immediate release type of tablet. And if it is not written as a tablet only, that is tablet extended release, then it will be a extended release type of formulation. Here sitagliptin phosphate and simvastatin tablet paddle with stainless steel stationary uh, uh, basket type of holder, or the additional requirement is given here. Then speed is given, media is given, volume is given, and the time points are given. So by this way you can search the top formulation of your interest. I hope this video will help you to use the database and understand the dissolution method. Thank you for watching the video, and I request you to do like, share and subscribe to this channel. Thank you once again.